Thanks, Judy, for a great talk. So next we've got Professor Rob Bristow. So Rob is the lead for cancer immunology. Rob is a clinician scientist who joined Manchester last year in 2017 after working in Toronto, so at the University of Toronto, but also at the Princess Margaret Cancer Centre. So this is one of the largest cancer centres in the world. And during his time in Toronto, Rob was twice awarded the Canadian Foundation for Innovation Award. Rob is now the director of the Manchester Cancer Research Centre and is the chief academic officer at the Christie. And he has a special interest in prostate cancer and specifically tumour hypoxia in clinical trials. So Rob's going to talk to us today about the research taking place within the cancer immunology branch.
Thank you very much for that introduction. So I'm a bit of an imposter as well, because I'm not a card-carrying immunologist, but I want to be. And when you want to be within a cool family, and I think I found one. And so what I really want to talk about is the ambition of the Manchester Cancer Research Centre to have a robust cancer immunology program that has a different USP from others. And that means that we really have to go back into the basic science and work with investigators and the Lydia Becker to really understand some of the fundamental reasons why, in some patients, immunotherapy doesn't work, despite the great, press that immunology and cancer immunology has. At the end of the day, immunotherapy is complicated, it's very expensive, it has severe side effects, and we really have to understand the basics of immunology when we come back and rethink why patients aren't responding. So I think at least there's a very exciting time, of course, when we think about checkpoint inhibitors and CAR T-cell therapies within the cancer realm. And I think also what you're going to hear from me today is that recent success in an iMatch grant, so this is a networking grant from Innovate UK, allows Manchester to take a real step forward in real-world trials in immunotherapy and actually to get them done, to understand a lot of the data in terms of patient responders and non-responders and also side effects. And when we've done really an environmental scan in Manchester in collaboration with Tracy and other colleagues at the Lydia Becker, I think there are a number of themes that we will work on in Manchester to really drive our USP forward in the cancer immunology space. And I've listed them here. The first is the patient specificity in terms of these responses, and with an overlay of comorbidity. We have complex patients in Manchester, and we shouldn't just be focusing on cancer. We should be focusing on cancer in the context of COPD, in the context of diabetes, in the context of cardiac disease, and there will be different immunobiologies associated with each of those disease states. So the understanding of that complexity, I think, allows us a way forward. The other issue, of course, is to understand inflammation in the context of early lesions, in terms of the early detection of cancer. And again, how can we pull apart signals that might come from certain morbid states, that of early cancers. And finally, two other areas, which are the predictive assays for patients who are receiving immunotherapy after $1 billion worth of investment in many checkpoint inhibitor trials. At the end of the day, we don't have a robust biomarker for an individual patient coming through the clinic as to whether there'll be a responder or non-responder. And so therefore combination therapies that use radiotherapy plus immunotherapy, chemotherapy, immunotherapy, we need to understand the basis of those standard of care treatments in immunolandscape and then the additional impact of immunotherapy. So when I came to Manchester and became the director of the Manchester Cancer Research Centre, I brought an ethos which was cancer team science. And you see the definition really here, which is deep integrated knowledge, potentially of disparate groups. I think that's true in Manchester. We're down the road a little bit from the Lydia Becker, but we find it very easy to come uptown to interact. But also that you have an alignment, a goal alignment. And so one of the things we've been doing with the Lydia Becker is to really say, what are the important clinical questions, the clinical areas that we can work on together, and that usually involves a gin and tonic. So when we wanted to really think about the cancer research community in immunology in Manchester, we started to actually get our act together on the cancer side, which is to bring like-minded investigators together who potentially were siloed, not for any other reason other than they were growing their own programs, but there wasn't a platform to interact. So the Manchester Immuno-Oncology Network has now been formed. You can see the aim. And actually, we already have had 2 workshops, on to our 3rd workshop, looking at novel preclinical models, again, for example, in a in a comorbidity approach, and really driving forward an understanding and a conduit from that preclinical data to the clinical trials. And again, trying to really think about how we can effectively link to the Lydia Becker with this approach. And the reason it's very simple is that when we think about T-cell responses and blockage of those responses and then inhibitors that really can take a tumor that is relatively cold, quote unquote, to a hot state, at the end of the day, as I said, for an individual patient, we don't know the predictor. And this is just some data, again, from the Pacific trial, a trial that has got a lot of press recently in terms of adjuvant therapy with checkpoint inhibitors after chemoradiotherapy for lung cancer. And you can see that those curves are quite disparate with respect to the addition of adjuvant PD-1 inhibitor. The reality is this also now is not only transformed our approach to lung cancer, but also it's improved now overall survival in a disease that otherwise had fairly dismal survivals, and that was just updated at the World Lung Cancer Congress last week. However, it's not completely curative, as you can see by those curves. So clearly there are responders and non-responders, even with the addition of this therapy. And so when we try to look at the entire area of PD-1 inhibitors, although the first trials were extremely exciting in melanoma and also in lung cancer, there are a number of drugs now that are out there. The choice of one versus the other is usually on the basis, of course, of the reality of the medical oncologist relationship to the companies. But how do we pick them more judiciously in terms of the combinations? And the other issue is, as I said, is that some patients don't show that long-lasting remission, and some patients have severe side effects. And so this really underscores a need in Manchester, where as we are adopting these therapies at a rapid rate, for us to really understand the patient-specific complexities and really roll that into our trials. So I'm just going to highlight a number of different programs. The people are in the audience. The posters are just outside. So Santiago Zeleny is part of the Cancer Research Manchester Institute, and really his group is really looking at this interplay between inflammation and cancer, and has really found some fundamental properties about our understanding about the types of cytokines that are involved in cancers that will go on to regress or will go on to progress, and also how can we actually inhibit some of those inflammatory processes to actually activate a better response to PD-1 inhibition. And so when we think of the dual role of inflammation in cancer in terms of immune infiltrate, I think Santi would argue that it depends, quote unquote, as to whether or not you actually have a tumor progression or tumor control. And it's that understanding of where that question mark is that his program is really driving forward for us to think about not only the immunolandscape and heterogeneity within tumors, but how do we actually harness that for differential treatments. And I think this was an important paper really looking at COX-2 inhibition in combination with PD-1 blockade, and showing that the animals that were treated with both, actually you can see this beautiful reduction in terms of tumor growth and almost really flatlining, suggesting a potential long-term cure. So the inhibition then of the inflammatory response, in addition to a PD-1 blockade here, is showing extremely good promise in the preclinical state. And so his lab is really trying to understand this interplay between inflammation and PD-1 checkpoint inhibitor responses, and really thinking about how can we better use anti-inflammatory drugs, non-steroidal anti-inflammatories, and COX-2 inhibitors within clinical trials, and has already started with Ann Armstrong, a clinical trial in this space, to really see if that preclinical data are going to hold up in patients. And therefore, understanding again the relative cytokines that might be cancer promoting, associated again with inflammatory infiltrates, versus those cytokines that would be cancer inhibiting, and then actually classifying cancers on the basis of their secretion of these cytokines and mRNA expression panels, and actually trying to come up with, again, a predictive assay as to which patients will actually respond with anti-inflammatory combinations. And we think this is so important that we started to say, well, you know, Santi is working in this area, we have a number of other people working in this area, why don't we actually come together with a collaborative project? And so actually this is one of the ways, of course, to stimulate the interaction is actually put in a grant together. So we have a very strong lung cancer screening program in Manchester, and actually we have low-dose CT scanners that go on vans to underserved areas and invite the public actually to have a lung health check. And in that case, we are screening for cancers and picking these cancers up very early. But what we realize is actually the immunolandscape of these cancers is not well understood. So with Tracy and others, we are actually now going to look densely, and I'll come back to the biomarker panel at the end, of the immunolandscape of these cancers, and I'm trying to understand and really pull apart the genomics of the cancer relative to the immune cell infiltrates and relative to inflammation. And so this is just a schema here of how we're going to put it all together, and what we really want is a multimodal biomarker that will tell us which patients are most at risk for cancer, and then to come in with a preventive strategy, and that might include, of course, now an anti-inflammatory approach. So the reality is that in Manchester, we have the best kitted out radiotherapy department in the United Kingdom. We have proton beam therapy, which is an elegant way, really, as a depth charge to place radiotherapy different parts of the body, particularly reducing long-term side effects in pediatric populations. We also have an MRI machine attached to a linear accelerator, an MRI LINAC, again, to achieve high doses with less side effects. So that precision, physical precision, has to be married to biologic precision in order for us to really have great impact. And we're really very curious then, how can we combine, for example, immunotherapy plus radiotherapy in the best context, again, to provide not only better local control for tumors, but also to have an effect on systemic therapy with respect to adjuvant treatments. And so Tim Illidge, who is also in the audience, is driving forward these studies on behalf of Manchester and is world-renowned for his work. So we're very interested in building syngeneic models, and Tim is doing this, to look at the different immunoresponses to radiotherapy alone, and then to radiotherapy with checkpoint inhibitors. Very interested in hypoxia, so decreased oxygen levels within cancers that have an adverse prognosis across every single tumor type, and what is the immune infiltrate in those areas relative to well-oxygenated areas. And then also protons versus photons. They have different physical characteristics. Do they have different characteristics with respect to immunophenotyping? And so the rationale for the combination approaches really is shown here, where as you go forward in terms of the different uses of immune checkpoint blockade, the concept is to use radiotherapy actually to activate the T-cell responses and to understand the cytokine releases, so that in fact a checkpoint inhibitor can be utilized in those tumors that are actually activated by radiotherapy. And we have no idea what the percentages are for those patients within different tumor types. So this is a very nice paper from Tim's group looking at overcoming the resistance to fractionated radiotherapy using concurrent PD-L1 blockade. And really the concept here is that that the activation of the response by radiotherapy can now be recognized and can be driven forward to an adaptive upregulation of PD-1, and also this piece of work links then CD8 T-cell production of interferon gamma dependency to actually incur tumor control, and I'll show you that data right now. So this is data now looking at non-treated tumors, or tumors that are treated with anti-PD-L1, or tumors that are treated with 5 doses of 2 gray of radiation, 2 gray being a clinically relevant dose. And you can see in this particular tumor model that Tim has shown that the combination actually leads to increased survival compared to any of the other treatments alone. What was also important is that in terms of an immune cell knockout approach, that Tim was able to show that these responses with the anti-PD-L1 were in fact CD8 T cell dependent, because when he knocked out CD8 T cells, he actually did not see the same response, and then again confirmed this on multi-parameter flow cytometry. And importantly, to think about clinical trials, when do you actually add a PD-1 inhibitor within the radiotherapy regimen? And the reality is, again, is you could think about doing it at the very beginning or in the middle of radiotherapy, or actually using it in an adjuvant setting, which many clinical trials are now using in medical oncology. And what Tim was able to show is the Schedule C, which is adding it after but not concurrent with radiotherapy, was not efficacious. So in fact, you have to have it during radiotherapy, and this has informed a number of trials now ongoing in Manchester and across the United Kingdom as to how to use radiotherapy in this setting. I think the other exciting area, but also very expensive area, is CAR T-cell therapy. And of course, the UK government has put quite a bit of interest in terms of activating this therapy for leukemias and lymphomas that are otherwise refractory in the pediatric and adult populations. And the concept of, again, that we can utilize and harness now molecular altered receptors for T-cell responses in patients who otherwise would have had lethal disease has led to, in this case, refractory B-cell lymphoma, long-term control. So much higher duration of responses than ever before seen. But this is an expensive therapy. It's £340,000 per patient. And so when we think about the use of this therapy, these results are very important, but we have to use it judiciously. And of course, can we think about now the use of CAR T-cell therapy across other solid tumors where it's already working, as I said, in relapsed or refractory large B-cell lymphomas or for acute lymphoblastic leukemia in children. So this is where Dr. Fiona Thistlethwaite is involved, and she is the director of a new program called iMatch. This is a networked program with 2 other centers in the United Kingdom funded by Innovate UK to actually put the practicality of cell therapy and gene therapy into practice and to look at patient-reported side effects and to also look at efficacy data. So at the end of the day, we really need to know in a complex multi-step process for actually, again, producing these medicines as well as the use of them, and then to find out who best benefits, you need a very slick operation, and that's what iMatch actually achieves or will achieve. So this was a great success for Manchester because not only did it bring together through the Manchester Cancer Research Centre a number of different academic sectors and trusts, but also it allowed at least 9 industrial partners to be part of an academic corporate group that is now leading this for the Manchester area. And so the vision is really to transform the conduct of advanced therapeutic medicinal projects, and it's only, as I said, one of 3 national centers with the objectives that you've seen before. Now, all of these things might seem quite simple in terms of sample traceability, et cetera, but when you're thinking of really building a program and viral production laboratories and getting that into trusts and already busy hospital settings, and then to actually have, again, all of those clinical elements that have to be there, including, for example, MRI for potential neurologic complications. What Fiona is doing with collaboration of her clinical colleagues is just making sure that this is going to be safe, effective, and done quickly as possible, and that also this can be used for a number of different tumor cell types. When we look at the trial portfolio right now in the early phase unit associated with the Christie Hospital, clearly there are a number of immunotherapy trials that are ongoing, and Elaine Kilgore now, as part of the clinical and experimental pharmacology group, is really driving forward studies in terms of finding the best biomarkers potentially associated with best responses of the patients. And so she and Caroline Dive has set up a tumor immunology and inflammation monitoring laboratory to actually do this in real time with patients who are on trials. And so this really is, again, will be a world-renowned laboratory in order to be able to look at a number of different endpoints for patients who are ongoing immunotherapy trials within Manchester. So just in terms of bloods and in terms of markers, we can look at immune cell subsets, cytokine storms, and also mutational burdens using circulating tumor DNA, as well as TCR clonality. And on tissues, furthermore, we can look at in situ infiltrates within the tumors, as well as immune gene expression on biopsies and surgical resections coming from patients in both FFP and frozen. So a real step forward in our ability to look at multiple endpoints and again come up with multimodal markers of responses. And just for example, Fiona has Thistlethwaite has a trial looking at tyrosine kinase inhibitor plus a checkpoint inhibitor in an adjuvant setting as part of a renal cell carcinoma trial. And you can see the type of dense biomarker analysis that our laboratory allows in terms of trying to again discern those patients who are responders and non-responders. And this is just a shout out to one of our Dean's Prize investigators, that's Rob Metcalf, who's a medical oncologist working with Tracy, really trying to understand the immunophenotyping of head and neck cancer, particularly salivary cancers, and again, with a defined protocol, really driving forward multi-TOF approaches to look at immune cell subsets, and again, in patients who are undergoing treatment and looking for resistance patterns in this particular regard. So I think there are some exciting strategic collaborations generations ahead. We certainly have a very strong approach now to ex vivo biomarker programs, and this is in collaboration with the Lydia Becker. I think what this affords is an ability to back translate what we're finding in the clinical trials back into model systems, and that's something that we urgently require and will be a collaboration with the cancer program as well as Lydia Becker. And I think the other is a real focus on inflammation and anti-inflammatory approaches in the patients that actually would benefit from this, and can we use it as a preventative strategy when we take that knowledge that is usually apportioned to late-stage disease and really start to collect earlier samples, and even in pre-malignant samples, to try to understand the immune subsets. And finally, we'll be a world lead in actually to look at what are standard of care therapies due to the immunolandscape of tumors, radiotherapy by itself or chemotherapy by itself. And with Tracy, we're driving forward again our understanding that maybe only 20% of patients are activated by radiotherapy, but those are the 20% of patients who would most benefit from a checkpoint inhibitor. And in fact, that's the biomarker, and you could take it at week 2. So we're very excited to collaborate. Tracy, lots of gin and tonics to come, I'm sure. And really, I can't say enough how the Lydia Becker has really uplifted a brand new cancer immunotherapy program citywide for the Manchester. It's just a pleasure to be up here and talk about it. Thank you.